for inviting me to participate in this virtual seminar series, in which I will be talking about a particular aspect of brain-body interaction that has attracted our attention for the last, I would say, 20 years, which is how different metabolic and nutritional signals participate in the brain control of reproductive function, and particularly the control of puberty. And in my presentation today, I will mainly show you some of the data that we have gathered during the last 5 years or so, or 10 years, I could say, on novel neuropeptide pathways and central energy sensors that we do believe participate in a very relevant manner in the metabolic control of puberty. So just to start with, I will show you first a schematic of the so-called hypothalamic pituitary gonal axis or reproductive axis. So this is a neuroendocrine system that we will be working, so to speak, during our presentation today, which is responsible for the control of reproduction and gonal function throughout or at different stages of the lifespan. So in this neuroendocrine system, as you can see here, you have different signals coming from different sources. The hypothalamus, where you find a prominent population of KIS1 neurons, or sorry, of GnRH neurons located in the basal forebrain, the pituitary gonadotropes that produce gonadotropins, and then the gonads. And these factors or these levels are connected through feed-forward and feedback regulatory loops in which the different signals, GnRH, gonadotropins, and hormonal comings from the gonads participate quite actively. And this is a very hierarchical neuroendocrine system, so that GnRH neurons in the hypothalamus are the major hierarchical hub responsible for integrating a wide diversity of signals coming from other parts of the brain or the periphery to very precisely control the functioning of this system throughout the lifespan. And already highlighted in this scheme, signals coming from different tissues of the body with a relevant metabolic function, such as, for example, the white adipose tissue, but also the gastrointestinal tract and others, like the liver, even the muscle or the bone, have been shown to closely interact with different levels of this hypothalamic pituitary gonal axis, mainly the brain, to precisely control reproductive function. And the same holds true for the particular transition that represents puberty. So puberty is a developmental continuum in which reproductive maturity is acquired, and this is defined by the full activation of the HPG axis that takes place during the pubertal transition. And this, in very brief manner, is brought about by the full activation of the neurosecretory activity of the GnRH neurons that I mentioned before, which in turn is regulated by the change in the balance between inhibitory signals and excitatory signals that converge on GnRH neurons. So actually, the timely activation of puberty is the result of the delicate dialogue between brain signals, mainly but not exclusively located in the hypothalamus, peripheral signals coming from different tissues of the body, including metabolic hormones, and environmental factors, which include, of course, nutrients, but also lights, other stressor, chemicals in the environment, etc., that interact to define the puberty timing to occur either in a proper manner or too early or too late. Why we are so much interested in studying puberty? Well, first of all, we are physiologists, and of course, puberty is one of the most fascinating developmental process in the body. And for this reason, we are very much interested in understanding how puberty is controlled. But we also believe that there are important translational implications for normal or perturbed puberty. And in fact, this is illustrated by the worrying trends of alterations in the age of puberty that have been detected both in the US and Europe, as shown in some of these papers here. And just to summarize the current state of the art, there seems to be quite consistent observation in which you see that there is an inverse correlation between the age of puberty that tends to go down and the prevalence of obesity, especially childhood obesity. So the more frequent childhood obesity is becoming, the more frequent is puberty happening a little bit too early. And this is interesting, as I said, in terms of translational medicine, not only because it defines a a phenomenon of unknown mechanisms in which metabolic signals are likely to participate, but also happens in a sexual dimorphic manner. So it's more common in girls, but probably happening also in boys. And very interestingly for us, it's bound to uncertain health consequences in the long term. So a number of studies, one of which I represent here in the slide, have pointed out a number of adverse health outcomes associated with either too early or too late puberty. So pubertal alterations are not only important in terms of reproductive maturation, but may also operate as health modifiers later in life. And for this reason, we want to understand why these changes in the trends of puberty are happening in different populations, including European populations, as I said before. And just as has been recently suggested for obesity itself, which has been proposed to be an hypothalamic-derived disease, we do believe that to a large extent, not only normal puberty is driven by the hypothalamus, but also alterations of the age of puberty are also related with perturbations of normal hypothalamic mechanisms. And that's why we are very much focused in studying these pathways that control normal puberty that can become deregulated in conditions of obesity. And in this particular context, a significant part of my presentation today will be devoted to this family of neuropeptides called kispeptins that I will present to you in a minute for those of you who are not familiar with them, that we believe are crucially important in the physiological control of puberty, but also in its modulation by metabolic signals and in parallel with its alterations in conditions of adverse nutrition, either subnutrition or obesity. So what are kispeptins? So kispeptins define a family of neuropeptides. And here you have the elements of this KIS1 or kispeptin system, also called the KIS1 GPR54 system, in which, as I said, a family of peptides of different length called globally kispeptin that can exist in different flavors if you want, with 54, 14, 13, or 10 amino acids, all derived from a single precursor that is encoded in mammals by a single gene that was discovered back in 1996 as a putative metastasis suppressor gene, and it was called KIS1. So it was in 2001 when the peptides encoded by this gene were characterized, and at that time it was discovered that they operate through a previously orphaned receptor called GPR54. So during my presentation today, I will refer to the gene as KIS1, the peptides or the ligands, the neuropeptides, are called kispeptins, and the receptor is called either GPR54 or KIS1R for kispeptin receptor. And I want to highlight that the initial discovery of the elements of this system was not at all related with reproduction. But all this changed quite dramatically in late 2003 because two independent papers documented that inactivating mutations of the kispeptin receptor GPR54 in humans and in rodents are bound to a profound state of central hypogonadism. In other words, reproductive failure of central origin. So these seminal observations highlighted the importance of kispeptins in the control of reproductive function, and particularly in puberty, as I will mention in a minute. But at that time, there was no knowledge at all on how these kispeptins could be operating to modulate reproductive function. And actually, I think that we can all agree as a community that in just more than, few years more than 15 years, so the time elapsed since late 2003 until today, kispeptin have been well characterized and are now regarded as probably the most prominent or one of the most important central regulators of the reproductive axis. We have described as a community the existence of discrete population of KIS1 neurons in the specific areas of the brain, particularly in the hypothalamus, where especially in rodents, but also in other species, there has been the reports describing the existence of two populations, one in the arcuate nucleus, the other one in the rostral hypothalamus. Both of them engage in the control of GnRH and are able to potently activate GnRH neurosecretion, and thereby they can conduct a powerful stimulatory action on the reproductive axis. So I think there is global consensus that indeed kispeptins play an essential role in the neuroendocrine control of GnRH secretion and thereby in the control of reproduction. How about the role of kispeptins in the control of puberty in particular? Well, a number of evidences have been accumulated during the last decade or so that have solidly documented the important role of kispeptin in the control or the modulation of the timing of puberty. I have tried to summarize kind of a long research or a wide diversity of research activity in a single slide that is shown here, in which I have validated a number of evidences that we believe demonstrate that kispeptins are important in the control of puberty. So the first came with the first characterization of the individuals, humans and mouse, with inactivating mutations of either the receptor or the gene encoding the ligands, the kisspeptins. And in all cases, there was a kind of conserved phenotype of lack of pubertal onset. In addition to that, initial studies, expression analysis, both in rodents and primates, demonstrated that during the pubertal transition, there is an increase in expression of the KIS1 gene, and also an increase in the sensitivity to the stimulatory actions of kisspeptin in terms of induction of gene rate secretion. And this seems to be caused by increased receptor coupling to the effector pathways, and this is modulated by a number of factors, including sex steroids coming from the ovaries and also metabolic hormones such as leptin that I will allude in a minute in a few slides ahead. So in general, there is good consensus that indeed kisspeptins are important in the control of the timing of puberty. And just to further stress on this, I show you a scheme that somehow is the summary of a number of studies that we conducted in our laboratory addressing female puberty in female rats or in rats as used as model species, in which we evaluated the external phenotypic index of puberty onset in rodents, which is canalization of the vagina. This operates as a biosensor of the increase of circulating estrogen levels happening during the pubertal transition. And I show you here the behavior or the normal progress of puberty that happens in control female rats. So you see that basically it's around day 34 that you see that this progression in terms of activation of the reproductive axis and canalization of the vagina is fully active. So completion of puberty usually happens before the age of 36 or so days postpartum. So what happened if in a set of animals, so actually what happens in terms of the hypothalamic kisspeptin tone? Again, to summarize a number of observations, we can schematize these as a transition between a state of low kisspeptin tone into a state of high kisspeptin tone that happens during this pubertal transition. So what happened in the group, if in a group of immature female rat, without any modification, you inject repeatedly kisspeptin into the brain twice a day between day 26 to day 31, 31, postpartum, you can advance vaginal opening and also advance other signs of puberty. So without any other change, you can induce earlier activation of the pubertal onset by just increasing the endogenous kisspeptin tone in the brain. On the contrary, if you block kisspeptin activity during this normal transition, what you get is the opposite. So evidence for delayed puberty, highlighting the importance of kisspeptin signaling as one of the players, not the only one, of course, but one of the players that are responsible for the normal timing of puberty. And in particular, we, as I will show you in a minute in the next slides, have been focusing mostly in this population of KIS1 neurons that in the recent years have become the center of attention for many neuroendocrinologists trying to characterize the neuroendocrine mechanisms controlling puberty onset. This is the population of KIS1 neurons, which is expressed or is located, in the arcuate nucleus, so the mediobasal hypothalamus, and this is important by a number of reasons. So in fact, the pulsatile release of GnRH seems to be driven to a large extent by the activity of this population of KIS1 neurons in the arc, but also very important for our presentation today, there is evidence suggesting that this arcuate population of KIS1 neuron, that is also called CANDI because they co-express other neuropeptides such as neurokinin B and dynorphin, is an important hub for the metabolic control of reproductive axis, and particularly for the metabolic control of puberty. And I will try to show you some examples on these during my presentation today. So again, trying to sum up a large body of evidence or experimental evidence, there have been a number of studies addressing the putative role of this ARC population of KIS1 neuron in mediating the actions of metabolic signals to control gene-rich neurons and thereby to control pubertal onset. And I show you here the particular example of leptin. Probably all of you know that leptin is a very relevant hormone in metabolic control. It's coming from the white adipose tissue and operates as a signal of adiposity or energy stores controlling different body functions. You need sufficient leptin stores or leptin levels to drive pubertal onset. So in absence of leptin, you don't go through puberty. And a number of studies have illustrated the potential role of leptin in modulating GnRH neurons via intermediate pathways, some of which go through the modulation of KIS1 neurons. So in brief, it's known that leptin can modulate GnRH activity, in other words, pubertal onset and reproductive function, in part by modulating the activity of KIS1 neurons. It's very possible that most of this modulation is indirect through this alternative pathway suggested here. And also it's true that there are leptin-mediated pathways that probably are not directly or primarily involving KIS1 neurons. But in fact, it's well known that conditions of energy deficit, such as, for example, subnutrition, in which leptin levels go down, in those conditions, you have low KIS1 gene expression or kispeptin content in the hypothalamus, whereas in the opposite situation, energy excess, too much leptin, such as, for example, obesity, you usually have some sort of biphasic response in which initially you have enhancement of the kispeptin tone, and this probably is associated with earlier puberty. And later, there is a switch into a kind of suppression so that continuous obesity causes suppression of the KIS1 system and central hypogonadism, especially in the male. So just to sum up what I have shown you so far, we could came to this kind of simplistic equation, if you want, in which the metabolic control of reproduction, and particularly puberty, is brought about the capacity of metabolic signals such as leptin to modulate the activity of KIS1 neurons, either directly or indirectly, to in turn regulate GnRH and thereby to activate or inactivate the reproductive axis. But again, of course, this is a very simplistic equation, and indeed, we are persuaded that there are important molecular elements incorporated in between these key hubs, leptin, kispeptins, GnRH, and actually part of the activities of our laboratory during the last decade or so have been directed towards characterization, the putative role of cellular energy sensors and mediators in this sort of framework, how the metabolic status of the body can control pubertal onset. So our original studies in this field date back to 2009, 2010, in which we provided the first evidence suggesting that one universal nutrient sensor of many cells, called mTOR or mammalian target of rapamycin, operating in the brain, plays an important role in mediating the permissive effects of leptin on KIS1 neurons. So to make a long story short, we provided evidence that if you block the activity of mTOR in the brain, this permissive action is lost, and thereby this causes suppression of KIS1 expression and prevention of normal pubertal progression. Later on, we were interested in trying to characterize the reciprocal dialogue between this nutrient sensor, mTOR, which becomes activated, for example, in conditions of energy abundance, such as, for example, those in which leptin levels are high. As I said, the potential interaction between mTOR and the genuine universal cell energy sensor called AMPK or AMP-activated protein kinase. So to some extent, AMPK and mTOR operate as functional antagonists. So for example, if mTOR is activated by conditions of energy excess, what activates AMPK is conditions of energy deficit. So for example, excess of leptin inhibits AMPK while it stimulates mTOR. So our studies were directed towards the hypothesis of whether this regulatory cassette in which AMPK and mTOR inhibit to each other could participate as mediator of the actions of nutritional signals, metabolic hormones, and other factors such as estrogens in the precise control of KIS1 activity and thereby pubertal timing. And to make a long story short, by a number of studies that have been published over the last few years, we have been able to come up to this working model in which we anticipate or assume that in conditions of energy sufficiency, so when nutrition is adequate, there is predominance on the activation of mTOR signaling pathways, such as, for example, in phase of high leptin levels, and this allows increased expression of KIS1 gene in the hypothalamus, particularly in the arcuate nucleus, and this allows pubertal progression in the normal process. In contrast, in conditions of energy deficit, there is predominance of activation of AMPK, and this happens, for example, when leptin levels are too low, and these cause decreased KIS1 gene expression in the arc and leads to pubertal arrest or delay. In the same context, we wanted to evaluate the potential role of another nutrient sensor that is participating in very many cells in controlling metabolism, which is SIRT1. So SIRT1 is a well-known member of the large family of sirtuins, as indicated in the scheme, that are involved in a wide diversity of biological functions, including the control of cellular metabolism and even the lifespan and healthy lifespan, if you want, acting among other sites in the brain. We were interested in SIRT1 and its putative role in pubertal control by a number of reasons, not only because it was known that SIRT1 could operate at multiple sites within the body and the brain to control very many different bodily functions, but also because of this sort of reciprocal stimulatory circle, whereby conditions such as, for example, calorie restrictions that are known to affect pubertal timing are able to bind both SIRT1 and AMPK in a dynamic manner. So in other words, we wanted to understand whether changes in SIRT1 or SIRT1 activity in the hypothalamus, and particularly in KIS1 neurons, could be a molecular hub whereby pubertal timing is modulated by nutritional cues. So we started these studies using mostly female rats or pubertal female rats as animal model by addressing changes in the expression of SIRT1 protein content in the mediobasal hypothalamus and the rostral hypothalamus during the pubertal transition. So I show you here for simplicity a comparison between the infantile period and the peripubertal period. And as you can see here, both in the rostral hypothalamus and in the mediobasal hypothalamus, where the arcuate nucleus is located, there is a significant drop in the content of SIRT1 protein during this pubertal transition. And interestingly, this drop in SIRT1 protein content was associated with a significant increase, as previously demonstrated, in the expression of KIS1 gene at these hypothalamic sites. What is the functional relevance of this change in terms of pubertal timing? To address that, we started with a relatively simple experiment in which we subjected the animals to moderate subnutrition. So the animals were subjected to a restriction of 30% daily food intake, so they're eating the 70% normal ration. And for this, this model of food nutrition, starting after weaning, is known, and we have validated that in our laboratory, to cause pubertal delay because of negative energy status. So as you can see here, vaginal opening as external sign of puberty is significantly delayed in animals suffering this model of subnutrition. And here you see the decrease in body weight. Again, this delay in vaginal opening was associated with other markers of pubertal activation. And interestingly for us, this was bound to a decrease in KIS1, which is an activatory factor, and this was associated with an increase in SIRT1 content. So in other words, there is a change in this reciprocal fluctuation between KIS1 gene expression and SIRT1 protein content in the hypothalamus of this subnutrition or these female rats subjected to subnutrition that were suffering delayed puberty. We wanted to see what happens if you just activate SIRT1 in the brain pharmacologically and without changing body weight in terms of pubertal timing. So we did the experiment using a specific SA3 SIRT1 activator, and we were pleased to find that actually this activation of SIRT1 in the brain without changing body weight was able to mimic the delay of pubertal onset caused by subnutrition. So as you can see here, there is a delay in vaginal opening. There is also delay in follicular maturation, or evaluated by histological means. And interestingly, this was associated also with a suppression of KIS1 gene expression, as indicated here. So altogether, I think that we have evidence for the existence of this sort of regulatory pathway whereby activation of SIRT1 is able to decrease KIS1 gene expression in the hypothalamus, particularly in the arcuate nucleus, and delay pubertal onset. But what happened in the opposite situation? This is overnutrition. So in this particular case, we use a model of overnutrition from early onset, meaning caused by combination of overfeeding during lactation, followed by high-fat diet after weaning, and by combination of these two obesogenic insults were able to induce a state of clear increase of body weight during the pubertal transition. So this increase in body weight is bound to a significant acceleration of pubertal onset as monitored by vaginal opening and also follicular maturation and other indices of pubertal activation of the reproductive axis. So this early onset obesity cause advanced puberty. But what happens in terms of SIRT1 and KIS1? So basically the expected reciprocal change. So these obese animals were having too low SIRT1 content and too high KIS1 gene expression. And we believe this was associated with the acceleration of puberty that we see in these obese female rats. And in fact, by looking specifically at KIS1 neurons in the ARC, we were able to document that in both conditions, overnutrition and subnutrition, there are congruent changes in the content of SIRT1 in specifically KIS1 neurons. So in models of overnutrition in which puberty is accelerated, the content of SIRT1 in KIS1 neurons is decreased, whereas in conditions of undernutrition in which puberty onset is delayed, happens the opposite. So the content of SIRT1 is increased. And this is compatible with our hypothesis that SIRT1 operates as a repressive signal on KIS1 expression to modulate pubertal timing. In fact, we further evaluated this repressive action by overexpressing using virogenetic means SIRT1 protein specifically in the ARC. And to make a long story short, by doing so efficiently using adeno-associated viruses stereotaxically injected bilaterally into the ARC, we're able to cause, without big changes in body weight, a significant delay in vaginal opening and the evidence of the first ovulation, suggesting again that when you enhance SIRT1 protein content in the ARC, presumably in KIS1 neurons, you get delayed puberty. To further characterize the molecular underpinning of this phenomenon, we conducted, together with Alejandro Lomnitsky in Portland, Oregon, a number of molecular studies, mostly chromatin immunoprecipitation assays, that allow us to propose the following molecular model for the action of SIRT1 in the nutritional control of puberty. So during normal puberty, this is the transition from the late juvenile period to the peripubertal period, basically you see a change, according to these CHIP assays, in the status of SIRT1 and other repressors of the activity of KIS1 gene promoter at the level of the KIS1 promoter. So before puberty onset, there is high presence of SIRT1 in the promoter, leading to a repressive configuration of the chromatin landscape at the promoter region. And this inhibitory complex is evicted during the normal transition of puberty, leading to activation of KIS1 gene expression and pubertal onset. In the case of in the case of obesity, already at the juvenile period, before proper pubertal onset, overnutrition caused this eviction of the repressive complex involving SIRT1. So this early eviction of this repressive complex allows an early permissive configuration of the chromatin landscape, earlier activation of KIS1 gene expression, and precocious puberty. In contrast, in conditions of subnutrition, this eviction that normally happens in the normally fed animal is not happening so much in the animal subjected to undernutrition. Because of this repressive configuration, KIS1 gene expression is repressed, and this leads to delayed puberty. So putting together all the data I have shared with you regarding our studies on cellular energy sensors, I think we have came up with this sort of conceptual framework, whereby by the action of specific cellular energy sensors, including phospho-AMPK, SIRT1, and mTOR, we can explain either the earlier activation of KIS1 neurons that happens in conditions of obesity leading to puberty onset, in which premature eviction of SIRT1 probably leads the process of accelerated puberty together with activation of mTOR, and also the opposite, delayed puberty caused by energy deficiency, in which you find activation of phospho-AMPK together with persistence of this repressive complex at the level of the KIS1 gene promoter. So in other words, I think that we have provided evidence on how different nutritional and metabolic cues can converge on these regulatory elements, mTOR, AMPK, and SIRT1, that are able either to activate or inactivate KIS1 neurons that also interact to each other, either with repressive or stimulatory loops, and thereby modulate by downstream effectors the activation of the reproductive axis at the time of puberty. And the final part of my talk, just the last 5 or 8 minutes, I could say, I will show you some of the data that we have collected more recently, trying to characterize the molecular nature of some of these downstream effectors that we believe are playing a very important role in the effective modulation by nutritional cues, particularly in conditions of obesity of pubertal onset. And in particular, I will talk to you about our recent work addressing the roles of brain ceramides in this process. I think I don't have to expand too much on the description of ceramides. This is a large family of lipid mediators that are generated by rather complex synthetic pathways and participate in a wide diversity of cellular functions. Interestingly, for our project, I want to highlight the importance of the so-called de novo ceramide synthesis pathway, in which ceryl palmitoyl transferase is a key initial step in the synthetic machinery leading to ceramides. And this de novo pathway can be either activated by administering the precursor THER6 or blocked by a pharmacological inhibitor of SPT that can block effective production of ceramides. In recent years, proper de novo ceramide signal synthesis in the brain have been pointed out as an important effector pathway for mediating the biological actions of a number of metabolic hormones, mostly leptin and ghrelin, in the control of different metabolic processes, including body weight homeostasis. So in this context, we became interested in evaluating whether central ceramide synthesis, and particularly de novo ceramide synthesis, could also participate in pubertal control, and particularly in its modulation by nutritional and metabolic status. So we started our studies again using this model of obese or early onset obesity in female rats, in which combination of two obesogenic insults, so rearing in small litters, in which you have more access to milk and then overnutrition, which is followed by high-fat diet, is able to cause a persistent increase in body weight, and this is bound to accelerated puberty. So earlier vaginal opening and earlier first estrus as a sign of first ovulation. So in these models of early onset obesity causing precocious puberty, we evaluated the content of ceramides, particularly in the mediobasal hypothalamus, and in line with previous reports in obese adult rodents, it was documented by our study that in collaboration with Núria Casals in Barcelona, that in fact these obese animals do have an elevation in the content of ceramides at the hypothalamic level. And it's not only that total ceramide content is elevated, by as much as 12 different ceramide species, as shown in the lower part of the slide, are elevated in our model of early onset obesity and early puberty. So early obesity can cause an elevation of ceramide levels in the brain and particularly in the hypothalamus. But what is the functional consequences of that? In our first approach, we undertook pharmacological experiments again, using the tools that I showed you before, either THER6 or myrcene. So when we infuse THER6 in the brain of immature female rats, we found that there was no change in body weight, but the sign of puberty, of the first sign of puberty, vaginal opening, was advanced, and the same happened with the marker, histological markers of follicular maturation. So there was clear advancement in the timing of puberty caused by enhancement of ceramide synthesis in the hypothalamus or in the brain. In the opposite, when we use myrcene to block de novo ceramide synthesis, again, we found no change in body weight, but vaginal opening was delayed and follicular maturation and first ovulation were also markedly delayed. So just by changing the content of ceramide in the brain, we're able, without changes in body weight, to modulate pubertal timing to cause either advancement of puberty by enhancement of ceramide content or delay of puberty by blockade of ceramide synthesis. So we became very much kind of excited with this finding and wanted to evaluate whether this ceramide signaling pathway could cross with the kisspeptin signaling pathway to modulate pubertal timing. So first experiment, again, was a pharmacological one in which we wanted to evaluate the effects of kisspeptin on pubertal onset and see whether these are dependent or not on proper de novo ceramide synthesis in the brain. So for that, we use a model of subnutrition again. With this model, you lower the endogenous kisspeptin tone. And by lowering the kisspeptin tone, here you have the control animals fed at libidum, female rats during pubertal transition, and this is vaginal opening. So by subnutrition of 25% in this case, you see that there is a significant delay in vaginal opening. So vaginal opening is almost totally prevented. So if you administer kisspeptin in the brain twice a day for 7 days around puberty, you can rescue to a large extent vaginal opening, meaning that pubertal activation is taking place. But if at the same time you block de novo ceramide synthesis by using myriosin, this effect is totally prevented. So in other words, in order to kisspeptin effects being detectable, or in order to kisspeptin effects in terms of pubertal progression happen, you need preserve central ceramide synthesis. So we wanted to evaluate whether this is related with the classical actions of kisspeptins that I showed you before are related with activation of GnRH neurons to activate gonadotropin secretion. And by a number of in vitro and in vivo studies, again, to make a long story short, came to the conclusion that despite the dependence of kisspeptin actions in terms of puberty on proper ceramide synthesis, ceramides apparently were not needed, or on the contrary, were totally dispensable for the actions of kisspeptin on its classical targets, which are GnRH and gonadotropins. And this, of course, suggested that the pubertal actions of ceramides are not happening through the classical or putative kisspeptin pathway involving the gonadotropic axis, but rather are complementary and happening through an as yet unknown alternative pathway. So we wanted to evaluate, or wanted to address, which could be this alternative pathway. And again, to make a long story short, we came across the potential role of the paraventricular nucleus, and particularly of a population of neurons in the paraventricular nucleus highly expressing the synthetic enzyme, or the key synthetic enzyme for ceramide production, which is cerulopalmitoyltransferase, that I will show you in a minute, we believe play an important role in the metabolic control of puberty. Why we became interested in this particular nucleus, the paraventricular nucleus? I could say for three reasons mainly. First, we're able to document that this nucleus, depicted here, is receiving a dense kisspeptin fiber innervation during the pubertal transition in the female rat. And the density of fibers changes during or in conditions of obesity, as shown here. So probably our interpretation is that at the same time you see an enhancement of KIS1 gene expression in this obese model, together with a depletion, probably because of high turnover of release of kisspeptin in obese individuals. The second reason was that in the very same PVN, paraventricular nucleus, we found a clear labeling of neurons expressing cerulopalmitoyltransferase, the synthetic enzyme for ceramides, the novel ceramide synthesis, and this expression was dramatically upregulated in conditions of early onset obesity bound to precocious puberty. And the third reason is because this PVN, this nucleus, is the origin of the alternative pathway innervating, or sympathetic pathway innervating directly the ovary, that has been previously suggested to play a role in the control of ovarian activation. So as I mentioned before, we obtain evidence suggesting that kisspeptin fibers and expression of cerulopalmitoyltransferase change in the PVN of obese rats, and also observed that there were indicators of precocious activation detected by a number of markers, such as noradrenaline content in the celiac ganglion and the ovary during this obesity-induced precocious puberty. So what we believe is happening is that the normal activation of this sympathetic output to the ovary that happens in lean female rodents during the pubertal transition is accelerated in our models of early onset obesity. How important this is in terms of ovarian modulation or ovarian function? Again, we conducted first a pharmacological study in which we block central de novo ceramide signaling in our model of obese rats. And just to drive you through the data, which is a bit complex in this slide, we observed, as expected, that these obese animals in blue are entering puberty earlier and also are having the first sign of ovulation earlier. And by blocking with myriosin central ceramide synthesis, you can partially rescue or restore or delay this acceleration of puberty. And the same happens with markers of sympathetic activity in the ovary that are suppressed by central blockade of ceramide synthesis. So one could combine both observations to suggest that blockade of central ceramide signaling could partially normalize puberty onset and sympathetic activity in conditions of obesity. But to obtain a kind of a direct proof of the importance of this signaling pathway, we conducted phylogenetic studies in which we use interference RNA in order to interfere the expression of cerlyl palmitoyltransferase, specifically in the PVN. So as you see here, by using stereotactic approaches, we injected adeno-associated viruses expressing an interference RNA against SPT. We were able to effectively block the expression of cerlyl palmitoyltransferase in the PVN, which blocks de novo ceramide synthesis. And by doing so, we were able to prevent the acceleration of puberty that happens in obese rats, whereas in these injected with the viruses, this acceleration of puberty is not happening, despite the fact that they were equally obese. So both vaginal opening and first estrus were prevented in terms of advancement by this virogenetic approach, leading to the final scheme in which we believe have found a novel signaling pathway starting from the PVN, in which ceramide synthetic neurons are able to activate the sympathetic output to the ovary under the influence of KIS1 neuronal afferents to modulate ovarian maturation and to cause precocious puberty in conditions of obesity. Just to finish up, some take-home messages. So I hope I have convinced you on the importance of the metabolic factors in the neuroendocrine control of puberty, and in this context of the key role of the hypothalamic KIS1 system in the control of puberty by a number of factors, including metabolic hormones, that operate very crucially on this hub of KIS1 or candy neurons located in the mediobasal hypothalamus. We do believe that central energy sensors operate as kind of mediators for the actions of these neurotransmitters, including kisspeptins, in the metabolic control of puberty. And again, here, the important role of mTOR, AMPK, and SIRT1 has been highlighted by a number of experimental studies. And particularly in the context of obesity, we believe this altered SIRT1 activity in KIS1 neurons seems to play a very dominant role. And finally, in our recent work, we have found out, the putative role of central ceramide signaling as novel mechanism for the control of puberty, and particularly in the alterations of puberty bound to obesity through this novel KIS1 PVN ceramide sympathetic nervous system pathway projecting directly to the ovary. I would like to finish up just by showing, the list of our collaborators. I have highlighted in yellow those members of my lab that have been actively participating in the research lines that I have shown you today, our collaborators and our funding agencies. And with this, I will finish my presentation, and I will be happy to take any question that you may have.
Fantastic. Thank you so much for your presentation, Professor. It was really amazing. It was, as Rita commented here, was a really nice example of how the brain interacts with the body. So it fits perfectly in our ideology, right? Okay. So I have tons of questions. So there's one question here by Rita. Really insightful work and presentation. Thank you so much. I was wondering if an early onset in puberty in your manipulations will result in defects in reproductive output later on, such as infertility or, to an extreme.
Okay, that's a very interesting question that we are also addressing. As I mentioned before, I mean, it's clear that when you have persistent energy deficit, it's not only that you have late onset of puberty, but also you suffer from reproductive deficits. In the case of obesity, which is the aspect that is more interesting for us nowadays, what you see is some sort of dual or biphasic effect. So at the beginning of this early onset obesity, actually you see signs, especially in the female, of early onset puberty, probably because during this initial phase, there are mechanisms that we still do not fully understand, but probably related with these nutrient sensors that I have shown you today that activate the expression of KIS1 in the neuronal population located in the arc. But if you maintain this obesity long enough in the time, actually what you see are signs of central hypogonadism. So there is a switch between an initial excitation or activation towards a long-lasting inhibition. And
Okay.
this happens in both sexes, but is possibly more evident in the male, in which is well known that long-term obesity is bound to central hypogonadism. And we have presented evidence suggesting that in these models of central hypogonadism or gonadal suppression due to long-term obesity, there is a suppression of the expression of the KIS1 gene in the mediobasal hypothalamus. So probably same neurons detecting different sorts of metabolic stressors are behaving differently depending on the time course.
Okay. Very nice. Okay. Let me see. We have some other questions here in our. Okay. We have a question from Azure Rose, and she asks. She says it's a beautiful work. Thank you. Can you
Thank you.
please elaborate what is the sensitivity range of the system? For example, how big the change in leptin should be so that it's detected?
That's a very relevant question again, and I mean, if we take in a kind of a broader perspective, this also changes depending on developmental period. So, for example, in the models that we have used in puberty, I I stress that we have mostly used female rodents because female puberty is certainly more sensitive to nutritional stress than male puberty, but male puberty is also sensitive to nutritional stressors, even though the nature of this impact is different. But if you focus on the female puberty, for example, models of subnutrition of about 20%, which leads to around 20% decrease in body weight gain, which is not that body weight gain is not happening, but it's happening at a lower magnitude, so to speak, are sufficient to cause delayed puberty, I mean, lower leptin levels, delayed puberty, and reduced KIS1 gene expression according to our studies. So, in principle, the system seems to be relatively sensitive, of course, if subnutrition is persistently applied. If you look at what happened in the adulthood, then probably you need higher levels of subnutrition, for example, in order to get reproductive arrest. So probably in this case, 20 to 25 subnutrition only cause after several weeks an alteration of ovarian cyclicity. So sensitivity could change depending on the stage of maturation of the reproductive axis.
So I guess you're going into one question that I have, and maybe I'll ask it, and then we
Okay,
go back to the questions that we have here.
perfect.
Because what I was, maybe you mentioned it, but I missed it. So kisspeptin acts on just the onset of puberty, or is it also required to maintain reproduction at a steady level throughout life? And then let's say later on in the entrance of menopause.
Okay. Yeah, this is something that I did not very much elaborate, but I am happy that you asked because I think it's important to stress. So the view is that kisspeptins are important for, I would say, every single facet of reproductive function all through the lifespan. So these KIS1 neuronal populations have been shown to participate in sexual differentiation of the brain. They are important on puberty onset, so there is kind of a switch on of the system during pubertal maturation. But for sure, if you block kisspeptin actions after puberty, you
Uh-huh.
have an inhibition of the activity of the axis. So they are important not only to activate the axis at puberty, but also to maintain reproductive function.
Okay.
So, for example, if you apply subnutrition or fasting, for example, after puberty, you suppress KIS1 expression and you block reproductive
I see.
function. About menopause is interesting because, of course, reproductive senescence is different between rodents and humans. But if you go to humans in which you have a proper menopause, what you see is that because kisspeptins are regulated by the negative feedback action of sex steroids coming from the ovary, so basically, they are responsible for the negative feedback. So
Okay.
estrogens can control gonadotropin secretion in part by suppressing KIS1 expression in the arc. So what is seeing is that in the menopause, in human menopause, you see that there is a primary failure of the ovary. You get rid of sex steroids coming from the ovary in the menopausal
Mm-hmm.
woman, and because of this lack of break, initially KIS1 gene expression goes up,
I see.
trying to compensate, and
Of
that's why
course.
probably menopausal women do have high levels of gonadotropins.
I see.
In the long term, this system fails. For example, what happened in rodents, and actually in reproductive senescence in rodents, what you see is a suppression of KIS1 activity in the hypothalamus. So it's kind of
Very interesting.
complex phenomenon because it involves sex steroids in one hand and aging in the other.
Yes, I see. That's very interesting. Okay, let me see. We have another question by Carlos Ribeiro. Fan- well, it changed place. Let me see. Fantastic talk. I was wondering if there is a non-metabolic input into the KISS system, which will modulate puberty transition, mainly also cognitive or sensory input.
Well, regarding cognitive inputs, I'm afraid I don't have any direct evidence. It's an interesting question. I really never thought about it. But if you think in terms of translation, it could be very important because, in fact, it's well known that the affective and cognitive
Yeah,
environment of a girl can have huge impact on pubertal
okay.
timing. So
That's
actually,
exactly. So you're going into a follow-up question on Carlos'
okay,
question, which is exactly about that. So if
yeah.
there is anything about incoming touch information or
Yeah.
something like that.
So this is an aspect that is very, very interesting. To my understanding or to my knowledge, it has not been so well, or it has not been properly covered so far. So far,
Okay.
as I said, it's well known that this sort of emotional and cognitive influences are very crucial in terms of human puberty. So, for example, monoparental families, girls are usually entering puberty earlier, so advanced maturation and so on. To what extent this could be affecting KIS1 neurons, uh, I do not know. I think it's an
Mm-hmm.
interesting way of looking at that. I can comment on other inputs that are non-nutritional. So it has been shown that, for example, Grithism, so light dark
Ah,
cycles can influence very dramatically the activity of KIS1 neurons, particularly in seasonal species. So the length of the day can influence the activity of KIS1 neurons. Sex steroids, of course, are very important non-nutritional influences. So in fact, it's supposed that, or it has been proposed that the rising levels of estrogens are acting as kind of activators of KIS1 neurons during pubertal transition, and even other forms of stress, such as glucocorticoid-mediated stress, can also impact on
oh,
on KIS1 neurons. And to this could come a little bit close to the question in which stressful conditions could influence kisspeptin activity and thereby modulate kisspeptin output in terms of puberty onset or later reproduction.
exactly. Wow, very interesting. Very, very interesting. I think we don't have any more questions.
Okay.
So I still have some questions, but I guess I will ask you in our Zoom meeting afterwards.
Okay.
So
Okay.
I would just like really to thank you again for joining us today, and
Okay.
also for the people who joined us. And if whoever is interested can join us by just following the link for Zoom, which I just posted. I posted in the chat. So once again, thank you all for joining us today, and I see you in a bit. In a bit.
Thank you.
Thank you, Manuel.
Okay. So I will close this and go to the Zoom chat. Okay.
Exactly.
Okay.
Exactly. Thank you so much.
Okay. Thanks. Bye.